I'd like to thank Rob again and Mark for the invitation, and it's a very nice lineup of speakers and lots of interesting focus on this field of interactome biology. And I'm going to talk about a slightly different angle on the theme and talk mostly about phosphoproteomics and what we are doing in the context of cancer and also normal signal transduction. And I'll just recap, although you've had an introduction, just recap some basic aspects of mass spectrometry as they apply to global analysis today, and then talk about stable isotope labeling with amino acids in cell culture, or SciLac, and how we are using that to look at to identify therapeutic targets in cancer and using it for pathway analysis, how we have used it to dissect signaling downstream of a novel cytokine, thymic stromal lymphopoietin, or TSLP, and then talk about some of our bioinformatics efforts, NetPath, HPRD, and Human Protein Pedia as community resources. And we are very happy that Mike Washburn is here, and it's because, in part because of his major efforts in the mud pit technology that there is no running away from the fact that LC-MS/MS is here to stay, and multidimensional chromatography, and coupled to high resolution mass spectrometry that's here today. I think we can talk about where we can go into the future. And this is what can be achieved in an average, at least in research labs where you have very nicely tuned mass spectrometers and bioinformatics efforts. This is what one can do today. And this was definitely not doable as recently as about 3 to 4 years ago, where today we are actually approaching transcriptomic and genomic types of analyses. You can actually attempt to analyze the whole proteome of a cell line or tissue by fractionating it. Key to mass spectrometry today and to going deeper into the analysis is being able to fractionate or enrich, and this theme will come over and over again. So this is just one way to do things. So your one sample, the entire proteome of a cell line or tissue, in one tube rapidly becomes 30 to 60 or 100 fractions. So that means that's 100 different experiments that can be done, and each fraction can then be analyzed by LC-MS/MS. And you may sample, this is just some rough numbers so you can think about this, millions of peptides in the experiment, and until recently I had this number, which was still an optimistic number, to about 2,000 to 3,000. But now some labs are showing that essentially you can achieve such really in-depth numbers where you could be wondering, well, am I talking about transcriptomic or proteomics? You can get quantitative measurements on almost 10,000 proteins today, depending on how detailed your other steps are. And that's why I've written some ranges. Sample prep and fractionation may take you a few days, and the mass spectrometry can take you anywhere from a week to 4 weeks or so to be achieving these kinds of depth. That means that mass spectrometer then will be dedicated for that one kind of analysis. And time for data analysis, it can actually take you, just like many people who got into the gene expression measurement early on, they would take months to analyze. Proteomics data is in that. It's really the early days of proteomics. And you can do a very, very nice experiments and keep mining the data for months. 1 month is just one guideline. But really, what is catching on is, and you've heard this over and over again from many different speakers, many of them have focused on label-free kind of methods, which is where the research is going. But I'll talk about other kinds of quantitative mass spectrometry where you essentially introduce a mass difference either in vivo or in vitro. I won't talk about most of them, but mainly focus on one method called SciLac that I developed when I was in Mathias Mann's lab in Denmark. And this really, if you are expecting the changes to be subtle, this is, I would say, one of the better methods because of many of the advantages. And basically, what ends up happening is you encode your mass difference into every peptide of every protein without doing any of the hard work or the chemistry. The cell is doing it for you. And you essentially you grow the cells in heavy amino acid containing medium, and you can use many different labels depending on which protease you're going to use. If you're going to use trypsin, you grow it in heavy arginine and lysine. And I think Jeff talked about many of his experiments where they've used SciLac, and you have a control situation, you have some activated state, you mix samples. This is just one way of doing things where you can run a 1D gel, you can do an in-solution digest, SCX fractionation, name it. But the advantage is from the MS data, you get the identification of the peptide, map it back to the protein, but then this is the finding the needle in a haystack, where the ratio of the peptide signals, that is in the MS scan, actually tells you what is it that you are looking for. And in many of the scenarios in signaling or in biomarkers, the kinds of situations that we are interested in is not the abundant background, which is no difference between the two states, but one case, 1 out of 1,000, out of 100, out of 10, or out of 1,000,000, depending on which biological context you're operating in, where you really want to zero in on differences at the MS level. So it's a very simple in vivo method. It doesn't require any extra processing steps. It's uniformly labeling of proteins, and you can choose amino acids. And we have developed a website, scilac.org, where you can go, and we try to keep it updated on many different papers that have come. Yes?
So on the bottom right there, are you, when you're doing those ratios, are you summing the intensity of all the different, like the C13 containing ones? Or are you just, say, taking the 540 divided by the 537, or do you sum 540, 541, 540?
So different softwares do it differently. We take the C13 containing one, and different people are still devising software. I think what we are really excited about is, and this is where the development has been the least, is for the data acquisition software to ignore the signals from most peptides in real time, which have equal abundance, and then specifically fragment this. This is something that, you know, we are yet to see. We are working with certain vendors, but they don't seem to realize that that is really the next level that will really revolutionize and save time in a given mass spec run. But that's unfortunately not there, although there is activity in that. We have now you can compare 5 different states at the same time by multiplexing different masses of these amino acids. So that's the background. You know, we can use SciLac as a way to do quantitative proteomics, but we want to chart interactomes. We want to look at pathways in cells, and how exactly can we start doing this? Well, one of the ways, and you heard about this, is you can determine ordering of the components, protein-protein interaction studies. You can determine the interactions. But this is something that Anne-Claude talked about, mapping of enzymes, kinases and phosphatase in her case, and also in the kinds of examples I'm going to give you, in signaling is a key step. And this is not an easy one, because kinase-substrate interactions are transient. Stoichiometry of phosphorylation is low, and I tell this to people that, you know, it's very, very easy to do a global analysis. It's almost that when we end up publishing papers, we are misleading many members of the community, because they take that a little too seriously and apply that to their favorite protein. Global proteomics is easy. When you're trying to find every site on your protein of interest, suddenly things are 10 to 100 times more difficult. So analytical methods to identify phosphorylation sites, even when you know they're there, they're not easy. You can do global analysis, but deep and comprehensive, that's a different story. And then finally, establishing a protein as a direct substrate is actually not trivial. And by definition, you cannot do that in the context of a cell, because in a cell, you have other intermediary pathways and proteins. So you have to choose a situation which is in a test tube, where you mix a kinase with this potential substrate. Well, you have proven this is a substrate, but now this is an in vitro system, and therefore it's prone to errors. It's not the gold standard. You go back to cell. It's a catch-22 of anything to do with these kinds of PTMs. I'll talk about how we are using these charting of pathways for personalized medicine, and there are a very small number of examples out there where it is possible to do some sort of a targeted therapy, and it's mainly known for certain cancers, and Gleevec was one of the early ones, where it is targeted against a kinase that's active in chronic myelogenous leukemia. And one can wonder, how is it that we are going to deliver on the promise of personalized medicine, especially in the context of cancer? How can we find the next Gleevec? That's the kinase inhibitor against BCR, against ABL. And the key is not in developing more of those small molecules. Many small molecules against most kinases are already being developed by the pharmaceutical industry. What we really need to find to deliver on the promise of this personalized medicine is, we want to find therapeutic targets in individuals with cancer. The small molecule is being developed, but we need to find which target is playing a role in the causation of cancer in which human being. And I'll illustrate this with our study on pancreatic cancer as a prototype. We are taking a proteomic approach. So for many of these small molecules that are being developed, they are against serine, threonine, or tyrosine kinases. And our goal is to identify activated tyrosine kinases in a subset of cancers. By comparing the phosphoproteome, we are doing this as proof of principle studies in cell lines. These are primary cell lines from patients with pancreatic cancer. These patients are long dead, but we have the cell lines where we can study. And our idea is that although they have the same label, to the pathologist, they're all pancreatic adenocarcinoma. But what's happening inside the cell in terms of which pathway is active is different. And we want to find those activated or hyperphosphorylated. A key feature of kinases is that when they're activated, they autophosphorylate. And our goal is to find these hyperphosphorylated kinases that are specific to cancer cells and then figure out what they're doing in those. And we are using the SILEC approach where we take pancreatic cancer cell lines, compare them to their normal pancreatic epithelial counterparts, and then look for this type of a signal where we see more phosphorylation in the pancreatic cancer cell line compared to a normal pancreatic epithelial cell. And many people forget that cancers are heterogeneous once they get a label. So we may think about breast cancer, well, we have the hereditary and sporadic, but all the sporadic ones are not the same. Here, this is just one way to profile the proteomes of these individuals with pancreatic cancer. On the left, HPNE and HPD are two normal pancreatic duct epithelial cell lines. All the others are from different individuals. And this is a Western blot, IP and Western with anti-phosphotyrosine. So we are looking at a snapshot of what is happening in terms of tyrosine kinase activation, the tyrosine kinases, and their substrates. First thing is, even a cursory look tells you they're different. We didn't need to sequence the genome to figure that out. And then this is one of the normals. Even the two normals are somewhat different. Some of them have bands, and that's all we can call. This is a low resolution method. It's a 1D gel. They have some bands that could be one protein or many proteins migrating at that position that are hyperphosphorylated. And then there are others where, remember, it's a tyrosine Western blot. There is no extra phosphotyrosine content in many of these cells. And some of them look somewhat like each other. In general, they are pretty different. And what we did was zeroed in on this one cell line, P196, as a prototype where we see robust activation of multiple different kinases and substrates. We don't know what they are, but that's where mass spectrometry is a great tool because it's an unbiased way to figure out what is it that we are dealing with. So we decided to compare this with this using SILAC. This was going in heavy medium, this was going in light medium, and we carried out enrichment with anti-phosphotyrosine antibody, purified the proteins, run them on a gel, cut all of the bands out, and did MS-MS. And these are three different panels that are put next to each other, showing that we saw this is the band from the pancreatic cancer cell line. This is its normal counterpart. We see increase of EGF receptor pathway substrate 8, and EGF receptor pathway substrate 15. This is heavy, light, heavy, light. So we have a control built in. That's what I meant by needle in a haystack, because in any of these kinds of affinity enrichment methods, and you've heard many of the speakers talk about this, there is a certain background. Here, the background is being used to compare to the activated state. So these three peaks are coming from the pancreatic cancer cell line that we want to study. These are the quantitation ratios for each one of those proteins. And what is basically turning out, and these are the ratios, I've colored the proteins that we identified as being more abundant in anti-phosphotyrosine immunoprecipitates in the pancreatic cancer cell line versus control. So you can see, starting from the most upstream kinase here, EGF receptor, we see many of the substrates in this pathway lighting up. So it seems that EGF receptor is probably the most upstream receptor that is active in these cells, and we went back and we used more conventional methods. Until now, we are using mass spectrometry. Now we would like to see what is happening to the protein levels and their phosphorylation status by ways that many biologists can interpret more intuitively. And what you see is this is EPS8 and different proteins. The top panel in each case shows phosphorylation in that cancer cell line, and it's more in every case. But what is most surprising here is that the EGF receptor, the protein level itself, is not changed. I'm saying surprising because often you find in the biomarker field, pathologists love to take antibodies and just throw it on their sections, immunohistochemistry type experiments, and say this is involved or overexpressed. What we are talking about is not overexpression as in HER2 gene amplification that happens in breast cancer. That is something that people have taken maybe too dearly to heart, and maybe it's sending most of us along the wrong track. What we are looking for is activation of molecules, activation of kinases. We don't need to invoke a change in protein levels. You can also have situations where protein abundance may be changed, but if the kinase activity doesn't change, it's not active. If at all, there is less EGF receptor in this cancer cell line than the control. But you see this abundant hyperphosphorylation. What we have not figured out in this case is why is that happening? You can have many different mechanisms. We have gone back to this cell line and sequenced the coding exons, and there is no mutation in EGF receptor, which would be the simplest explanation. So we don't know the cause. We may have increase in ligand. We may have mutation in some other proteins that control EGF receptor activity or something upstream of that. That means a phosphatase possibly that negatively regulates EGF receptor may be involved. We don't know the full answer, but we know that EGF receptor is involved. And when we go back to what we did was we took this cell line and many other cell lines from these patients, and we grew them as xenografts in mice. And initially, we were very disappointed because we pulled out EGF receptor, which has really, in some ways, you can say, beaten to death by many people, proteomics and otherwise. It's really a well-studied pathway. So we're disappointed. But the good news here was there are inhibitors, small molecule inhibitors like erlotinib, that are available that we could use to test to see whether this cell line is actually going to respond to inhibition. And this is showing staining with a different reagent now, a phospho-EGF receptor specific antibody. That's this brown color in this case shows positivity. This is one other cell line where we did not have any mass spec data that also shows staining. And these are two different cell lines, tumors derived from those two cell lines where we don't see any staining. And when we treated these mice bearing these tumors with erlotinib, what we can see is this is where I showed you the mass spec data. We see that the tumor regresses in that and this other cell line where we see positivity, but not in these two other ones. That means now we have a higher throughput way of, again, now we are lucky with one other reagent. We have a phospho-EGF receptor specific antibody. So we use mass spectrometer as a discovery tool. We figured out that EGF receptor is active here, and then we go on to use small molecules and other kinds of reagents to figure out whether they are going to respond or not, and whether we can find out if they have activation of EGF receptor in a high throughput fashion. We also had access to other xenografts that our collaborator had actually published. They had data on this where they were testing many different drugs on a panel of pancreatic cancer cell lines. And what they concluded from their studies was that EGF receptor inhibitors such as erlotinib have no effect in pancreatic cancer. We went back to the data that they had, and we noticed, this is basically their graph, one of them seemed to be responding to erlotinib, but in the context of the big picture, it was left out and it was ignored. We went back to sections that they had from each one of those tumors, and the one that showed this response was the one that showed positivity. That means now we have a predictor of potential response to a small molecule inhibitor which was hidden in the data. And there are many studies that major medical centers have done in the context of pancreatic cancer, and they continue to do that in the context of other cancers. What is really unfortunate is that if you're using a small molecule, shouldn't you be looking at its target and whether that target is active? Because if the denominator here is this big number, only a subset of which that respond to the small molecule, the whole stage is biased against you. You are going to conclude that your drug doesn't respond when reality, 100% of those cases are actually responding. And I think this is the kind of a paradigm shift that we need to have in the context of many of the kinases. And just to show you that this is not really peculiar to pancreatic cancer. There's nothing strange about pancreatic cancer. We are systematically studying many different cancers, esophageal cancer, gastric cancer, and breast cancer, and we essentially see the same thing. The top panel shows you, again, the same experiment now. Anti-phosphotyrosine Western blotting is just, we have one key, and we are trying to figure out how many different locks it can open. And you can see in many of them, you see no signal. That means there is no blatant upregulation of tyrosine kinase activity. But look at the others. And when you, this is a small set of triple-negative breast cancers for which they're really tough to treat, and we have no handle on them. And how do we define them? It's really amazing how in 2009, it is still by exclusion. The reason it's called triple negative is some of the early markers that were identified are absent on this. Not to say that when those markers are present, we have made such wonderful breakthroughs. That's estrogen receptor, progesterone receptor, and HER2, 3 markers. And that's always, we are talking about not the activation of those pathways, but the amount of protein that is encoded by those 3 genes. Here, they are negative for those, and yet you can see that they are heterogeneous even at that level, because these are all grouped together as one unit now. And you can see molecularly. And it is possible that in some other ones, it's a serine-threonine kinase mediated pathway that is active. So we are basically now taking this large panel of cell lines, and we are trying to find out what exactly is going on inside. I'll change gears and talk about our studies on a ligand or a cytokine-mediated signaling scenario. Thymic stromal lymphopoietin is novel. It's amazing. And when I tell you the signaling story, you'll realize we are calling it novel, although it was identified almost 10 years ago. It's because it's still not a famous cytokine and not much is really known. But what has really happened in the last 10 years since the cloning of the cytokine is that it is a central player in asthma and other kinds of allergy. When they were preparing a patent application, I was involved in cloning the receptor. I was almost laughing because the lawyers had put in everything, that this is going to do everything, including allergy, asthma. They just named every immune disorder. And I was laughing because basically these are templates. You can put any cytokine, any receptor, and it's the same canned text. About 10 years later, I was to be proved wrong from my skepticism, because this cytokine, in contrast to many other cytokines, is actually very important and a central player in asthma. People have shown that in many different mouse models and also human studies now. But not much is known about signaling. If we know what is happening intracellularly, we can use those signaling proteins as potential therapeutic targets. So what is TSLP and how does it signal? Well, we know about the receptor Sabinus. I cloned this in 2000, thymic stromal lymphopoietin receptor, TSLP receptor. TSLP engages IL-7 receptor alpha chain, which is shared between IL-7, which is a more famous cytokine. That's why we still call it relatively new, although it's 10 years old. It uses this combination of subunits when IL-7 uses this. And basically, this is what was known in 2000. And in 9 years, the amount of progress that we have made as a community. And it also talks about the sociology of science. Who studies these types of cytokines in those kinds of cellular immunology assays? They're generally not biochemists. And also the tools are only now becoming very powerful to be able to do this. In 9 years, we have figured out that TSLP phosphorylates STAT5, transcription factor, ERK, and AKT. That's it. And there are lots of big holes. And what is known for IL-7, but many other members of the IL-7 family, of which interleukin-2 is a prototype, is that JAK family kinases are central molecules that take the signal from the juxtamembrane domain, go through STATs and other molecules. All of those were black holes here after 9 years of research. And in fact, what is amazing is in this particular case, because people are still keen on using the tools that they had, JAK family kinases were shown by many groups, stated in print that they are not activated. That data was based on Western blotting with antiphosphatase antibody, where you enrich those JAK kinases. It's a standard assay. Every biochemist uses it. So you enrich for those proteins, and you Western blot with antiphosphatase, and you don't see any difference. You say it's not activated. And then, but the scientists now say it's not the JAK family kinase that is responsible for transmitting the signal. We should try to find those kinases. And we are also very active. We thought, wow, this is a great chance at trying to do something major for biology. So what happens to the other siblings of this TSLP IL-7 family? This is the most well-studied protein in that family, interleukin-2 signaling, and it shows this is what we know. JAK1 and JAK3 kinases transmit the signal through STAT5, STAT3, and all these other kinases. This is what is known after many decades of research on this really a prototypical cytokine, IL-2. This is what we see when we started to do the biochemistry. And I think in the context of protein interaction, you have already seen at least some pictures like this, where if we activate cells with TSLP and use the tools that we have, antiphosphatase antibody, we basically see no difference. Now, does that mean there is no difference? Well, we don't know. But when you do another kind of an assay, once you know what molecule you're looking for, look at this here. This is enrich, that means immunoprecipitate STAT5, and you can detect it. Now you ask the question, is STAT5 tyrosine phosphorylate? The answer is yes. But it's just hidden behind the bands. It's a less abundant molecule that is getting phosphorylated. But if you had to just go by the sea of signal, you're not going to see it. So in that background, what we can now do is we can use SciLac. That's the power of this quantitative proteomics. When apparently you don't see any signal, you can go in and try to figure out in one step what are the kinases and the pathways that are being activated by this. And here, basically, we take a cell line that is a reporter cell line that responds to TSLP, and we split them into two. One, we start growing in heavy amino acid containing medium. All the proteins are labeled. Then we mix them, because we always know that the heavy peptides are coming from the TSLP treated cells. They're the same cell. And now we can take this very complex peptide mixture, the millions of peptides that we are talking about, and we can do many different things. And now, the previous experiments I showed you, we were doing enrichment at the level of proteins. Now, technology, this is 1 or 2 years into the future. Now I'm going to show you data where we are doing the capture of phosphopeptides. We have already done the digest here. All those peptides, of which most of them are non-phosphorylated, are there, and we are going to capture antiphosphatase peptides using an antibody. And on the other side, we are going to use either STX chromatography alone or in combination with titanium dioxide. And I'll show you just some representative data. All this is all unpublished and is very recent. So we are going to capture the phosphatase peptides from an antibody IP. And this is other kinds of enrichment principle. And our goal, our objective was to find a candidate kinase. We wanted, we knew it was not going to be JAK2. And this is a partial list. So we found hundreds of peptides, phosphorylated peptides, tyrosine phosphorylate, and I'm showing you the tyrosine phosphorylated peptides here, and the ratios. And most of them don't change. Because just for the cell, when it's not activated by TSLP, these are still getting phosphorylated for the normal growth. But we don't care about those. We want to figure out what are the kinases and their substrate that TSLP is going to induce. And this is what we find. So if we just look at the top two candidates, STAT5A and STAT5B, you see that, wow, we are on the right track. That already tells us. If you look at the bottom, this is just a small collection. We have two, but most peptides are in that group, CDK3 and GSK3 alpha. This is a doubly phosphorylated, this singly phosphorylated, this 1-to-1 ratio. And we have found many different adapter proteins and tyrosine kinases that are getting induced when you add TSLP. And until now, a lot of the data that you may see from biological experiments is usually dependent on Western blot with anti-phosphotyrosine, which is not strictly linear. So these numbers may not make intuitive sense to you, but these, which are amplified signals on the substrates of kinases, STAT5, a molecule, is between 3 and 10. So, and let's look at this JAK2. We found 1.8-fold difference on this site and 1.4 there. And look at LIN, which is another tyrosine kinase. This one peptide is actually downregulated, and a different site is upregulated. So we have found biochemical evidence by quantitative mass spectrometry that JAK2 is phosphorylated. We went back and redid the Western blots one more time. You cannot catch this difference. That 1.8 to 2-fold, you cannot catch it. And we have repeated the experiment, we find the same thing. We have gone on to do functional studies now. We have used JAK2 kinase inhibitors, and we are now doing siRNA-mediated experiments to show that what is driving proliferation in these cells is JAK2. And this is 10 nanomolar, which is very good for this inhibitor that we use in these studies. LIN, where I pointed out that we captured 2 different phosphorylation sites. One of them was at the C-terminus, and this is the picture that we see. This is uninduced, and this is TSLP-induced. This is reminiscent of what is seen in Src kinase, which is a prototypical member of this family, where the C-terminal tyrosine is an inhibitory tyrosine. For that kinase to get active, the C-terminal phosphorylation is suppressed or is dephosphorylated. So we see dephosphorylation of this, and we see hyperphosphorylation of that. This peptide comes from that. This peptide is here. So LIN is another tyrosine kinase that's getting active. And BTK is another tyrosine kinase that we've captured in that one experiment, single experiment. We have also, these are initial data again, and these are some of the data that we have from looking at serine-threonine phosphorylation triggered by TSLP. And now we're talking about a different class of proteins and different magnitude of change. Tyrosine phosphorylation was more subtle. Here we are seeing differences at the level of that phosphopeptide. The ones in red are the ones that we found by MS to be phosphorylated 10-fold, 4-fold, and again, hundreds of them were not changed. So the majority of them are unchanged, and we are trying to put all of this data together in some sort of a pathway picture. But most of these cytokines, the major action is through tyrosine kinase, and we believe that we have already found them. This was the picture of TSLP signaling when we began, and this is after many decades of studies. These are the critical molecules in IL-2 signaling. This is summarizing the data generated from many different platforms, many different labs over 2 decades. Today, this is where we are. It's not too bad for TSLP, where we still are yet to connect them. That's where we have to go back to the drawing board, and to be able to connect them and to find other molecules is going to take a lot more time. But we have already found many of these molecules, and we can now carry out functional studies using inhibitors against JAK2 and even LIN and BTK and TEC and other kinases for which we didn't have biochemical evidence. And the lesson that we learned, and I think maybe early on experimentalists are taught this, that when you don't see something, you just say it was not detected as opposed to it's not happening. And that's really the mistake we have made. Because every analytical platform has its own weaknesses, we have to be careful in negative data. If people had said not detectable, which is really what we can say, because somebody can come with another condition under which you will see activation. So here, that's one of the reasons why maybe the field got to be misled. And it ties in with other kinds of assays that others have talked about. You can have multiple reaction monitoring now, where you can just analyze only that one side. People shouldn't need to go through this entire proteomics experiment to monitor activation of JAK kinase. You can really monitor what fold activation you're seeing at the level of phosphorylation site abundance. And it's possible to take unbiased discovery approaches like this to dissect signaling pathways in a global fashion. I'm going to show you a few slides. It's an adapter molecule that I found in a proteomics screen when I was in Odense. I called it Odin, as an adapter molecule that was phosphorylated by addition of EGF to HeLa cells. And we have data from knockout mouse that this is a negative regulator of proliferation, but we really don't know how it works. And we flag tagged it, expressed it in 293 cells, and these are the interactors that we pulled out, where we basically saw no signal from the control cells versus Odin transfected cells. It's a long list, and I'll just show you what was known or what is known about this is also unpublished, interaction network of Odin. And we are hoping that this will shed light and we can figure out what is it doing, how is it negatively regulating EGF receptor signaling, but this is what was known. It binds, it's been shown in another higher throughput screen to bind to one of the members of a 14-3-3 proteins, and I'm now going to convert that long table that I showed you into this. There are many different proteins. Some of them are adapter proteins. This is a GTPase activating protein. This is a RAS-like protein. People don't know what that protein does. Some cytoskeletal proteins and enzymes. In that one single SciLac experiment, we have filled in all of the blanks, but this only sets the stage for doing many other experiments. And these proteins were already connected, and in this Odin IP, we basically identified all of those three. And you've seen this kind of a theme occur repeatedly through many of the talks today. So we are now working on, we have validated many of these types of interactions and are moving on. So this is great. We have no doubt that we can publish all of this data, but the problem is, although we were careful in going back, Odin goes by many different names. Many of them are, call it a KIA protein 0226. It's just an accession number. Now, how do we figure out what is the published literature on Odin? It's nice that we can go into supplementary tables and figure out, but that's really not easy for most people to do. And we would like to share our data, but also make it easy for people to be able to figure out when their findings are novel and what exactly is new. And you asked the question about this literature search. And I think it's a big one, because many of the experimentalists, they are dying to conclude the day the results come out, whether what they have found is novel or not. The problem is, some time ago, maybe people were more cautious, and they would say things like, to the best of our knowledge, this is novel. But now, because we have to be more and more aggressive, we have to sell our stories so much, we would like to just say, no, this is novel. But we don't know. There's so much of literature out there. Most of it is hidden in the supplementary pages. We would like to make sure that other people don't spend their valuable dollars rediscovering these things, or they take this to the next step. Because we cannot pursue many of these kinds of things. In the context of TSLP, what I showed you, in the context of pancreatic cancer, many of these pathways. It is experimental data, and we would like people to be able to benefit. So I'll tell you some of the mini stories about some of the thankless things that we are doing. It's really, they are definitely underappreciated at this point of time, and maybe in this room, we have a critical concentration of people who might appreciate these kinds of resources more than average. But still, I think there is a lack of appreciation for what we need to make systems biology happen. And this is one of the examples that I'm going to give you, NetPath, which is a public resource of signaling pathways that we have started to develop, because very quickly we have come from single genes to pathway analysis, and we would like to be able to say which pathway is activated, and this is our modest attempt. And our initial goal is to only work on ligand receptor type of events that can be clearly demarcated as a pathway, and to have a detailed collection of all published reports of pathway reactions. And initially, we have annotated them, the pathways that have more relevance to cancer and immunology, and to involve the community. And TNF pathway, many of you, I think there were some biomarker studies about TNF that were mentioned, but really when you go back to all that has been published on such a well-studied cytokine, it's something like this. It's almost unmanageable even when you zoom in. There are so many molecules that have been shown to be in this pathway that have talks and crosstalks in so many ways that to think that TNF receptor signal, and this is all the data that is published up to now. There are other studies that I'm aware of where 1,000 more proteins are going to be added to this pathway. 1,000. We should not simplify biology any simpler than it is. It is complex. And we need to try to understand that biology. And this is how detailed some of the pathways are, all derived from experimental data. Things are annotated in this map only when they change when cells that express TNF receptors are given TNF. That's the context. Yeast to hybrid kind of experiments are not here. TNF has to perturb the system. This is how detailed it can be. So this is a snapshot of the home page of NetPath, where we have done some cancer and immune signaling pathways. And the idea here is not just the physical interactions and the kinase and other enzymes and their downstream reactions, but also to compile a list of genes, downstream genes, that are differentially modulated by these extracellular stimuli, and also protein translocation events. I'll just walk you through some of these pictures. So this is EGF receptor. This is how many molecules. 177 molecules as of the date of this curation were already described to be downstream of EGF receptor. So we think it's only 20 molecules. That's a fallacy that we have. It's not what biology is trying to tell us. And then we have experts. This is physical interaction category. Catalysts, we have detailed comments written by curators, and transport, where when you add EGF, these proteins, they translocate from one compartment of the cell to another. And these are transcriptionally upregulated genes. So that's pathways. And that we have built on top of this research that we began almost 7 years ago as a non-redundant database for human proteins. That's the focus, human proteins. And our goal was to have a repository which would be a comprehensive repository of protein interactions and other special features of proteins like post-translational modifications, subcellular expression, tissue expression, and enzyme-substrate relationship. And when we began this, and even today, what is very interesting is for many protein interaction repositories, kinase, substrate, and these kinds of enzymatic relationships are not considered or classified under protein interactions, although they are definitely some of the most exciting interactions that you can have in a cell. This is the homepage. This is a molecule page in HPRD. If you are already not familiar with this, these circles are phosphorylation sites. These are protease cleavage events. And what we realize after many years of curation is that we cannot keep up with all the literature, that we must involve the community directly. And we started an initiative called Human Proteinpedia, where experimentalists could contribute their data directly, either through web interfaces or by interacting with us. And here you can see that this is the kind of metadata that can be captured from a mass spectrometry experiment. And then it is also available if people have contributed their data that goes along with that experiment. You can even inspect the MSMS spectrum in this case. And for this, I have to thank the proteomics community that really came along to help us. And Rob actually worked with us. He was one of the very enthusiastic participants to contribute data in this. And the NetPath that I described was done in collaboration with Gary Bader, who's also here. And these are the people in my lab who contributed to the experimental things. And this is the nonprofit Institute of Bioinformatics that I founded 7 years ago in Bangalore. And that name itself is a misnomer, really, because it is a systems biology institute. Institute of Systems Biology was just established in 2000, and this was founded in 2002. So that name, it was a very catchy name, was already taken. And what we said was, initially, we are going to develop databases, and we are going to focus on bioinformatics with a clear vision that we want to do everything. And now we have a genomics lab, so they do a lot of microarray type experiments, and they are doing a lot of mass spectrometry now. And we are very excited that they're joining hands with not just our lab, but many other groups in different places to do many of the thankless things that I think are really needed. But I think the community has still not come in ways that it should. We are all focused on data generation and not so much on data sharing, but I think that's really a weakness that will be fixed hopefully in the years to come. And thank you.
The question I had was, what kind of reagents you used for enhancing or increasing the phospho-serine and threonine peptides in a mass spec? In one of the data you showed that when you looked at your phospho-serine peptides. How did you actually?
Yes, so if you take some of the early fractions that come out of SCX columns, they are enriched in serine, threonine, basically all phosphopeptide species. And because serine, threonine phosphorylation is abundant, those fractions are automatically enriched. And then we use titanium dioxide to enrich. And you're right, I mean, the game is enrichment. If you don't enrich, you are going to miss many, many of those events. And we are still looking at the tip of the iceberg. So you can take those SCX fractions, then go do titanium dioxide on each fraction. So it's a lot of experiments, but that's the only way to go deeper.
That you mentioned showed prior to titanium dioxide, how many fractions?
Yes, so we are doing about 50, 60 fractions in all of those cases. The initial ones we analyzed without even titanium dioxide in the first pass, which is what has been done so far. So the titanium dioxide part has not even been done for those. And so what I'm showing you is some things that you're already finding to be regulated. So we have probably identified about 500 or so sites, and those, some of them are very nice adapter type molecules that we can find in that fraction.